Thank you all for joining us today. I'm Tracy Smith, the Program Director of Education and Outreach for the Center for Integrated Cellular Analysis based in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we will discuss new work exploring cellular dynamics during mammalian aging. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. So our guest today is Zehao Zhang, a graduate student in the Cow Lab at Rockefeller University. Zehao received his Bachelor of Engineering from Zhao University. His research focuses on characterizing organismal cellular responses to natural aging and aging interventions with novel single-cell genomics tools. So with that, welcome. I'll hand it over to you, Zehao.
Thank you. Thank you for the kind introduction and efforts for pronouncing my name. Can you see my slide?
That looks great.
Okay. Sure. So I'm gonna start. Hi everyone. My name is Zehao Zhang. I'm a graduate student from Rockefeller University, and today I'm gonna present a work that I've been working on in the past 3 years about dissecting the cellular architecture of the organismal aging. We are facing a relatively aging world by the projection of the United Nations. By 2050, there's a 20% of the world population gonna be over 60 years old. At the same time, aging poses a very severe risk for a lot of incidents of the aging-associated diseases such as cardiovascular disease, dementia, and cancer. And over the years, people have already trying to dissect the aging at the molecular levels, and they present some aging hallmarks. However, aging at the same time lead to the various physiological changes in different organs, and different organs response to the aging at a different level. There is a gap between this molecular level hallmarks and the organ specific response to aging. Is how the heterogeneous cell population exhibit aging associated molecular changes differently. And we really need to understand this aging hallmarks at the cell type specific level, which is the key for us to develop a precise aging intervention tools to achieve the healthy aging. And over the years, people already put a lot of effort to profiling the aging at the organismal scales. However, there's still some limitations. For example, in the published single-cell atlas on aging, they normally use the single-cell dissociation method to dissociate single cell for different organs. But this poses different challenges. For example, most of the single-cell dissociation have to dissociate cell at a 37-degree, which lead to the heat shock to the transcriptome make changes. At the same time, the single-cell dissociation method really need a fresh tissue, which lead to some problem for collaboration between an entire team to profile these different organs. And different enzyme dissociation really introduce the batch, and which is hard for us to compare the same cell type between the different organs. And also we hear from some adipose scientists at Rockefeller University that most of the single cell based method is not feasible to recover any adipocyte because the adipocyte is so fragile, and during the single cell dissociation, this cell just break, and which lead to the problem of the bias towards a specific cell type that is more easily to be dissociated. And most of this enzyme dissociation method really require the batch correction between the different experiment or between different organs, which lead to the potentially mask of the molecular features. The second problem of the current single-cell organismal scale profiling is that in the 2 or 3 years ago, there is still a big cost for like preparing a single-cell library. And so that's why like a private analyzing effort relatively profile a low cell number, but this really lead to the challenging for identifying the rare or like novel aging-associated cell types, and also is make it more challenging to use as a reference for the future single-cell studies. And the third challenge is like the private efforts still due to the like a high cost. There's like no lack of the replicate, not only within the same age, but also between the different sexes, which pose a challenging for like identifying the sex difference or like testing for the significance. And for my project, I'm trying to establishing an organismal scale profiling platform that tackle all these challenges. And luckily, when I joined the lab, our lab developed this EasySky platform that is a combinatorial indexing based platform that can profile this single cell in a very high throughput and low cost ways. And two unique advantages for this technique is that you can prepare a library for 1 million cells for only $700, and also our technique is not like a 3' end biased. Instead, it covers the full gene body, and which gave us both gene count information and exon count information. However, once I joined the lab, I trying to apply the EasySky to profile the different organs of the mammalian system. I still find this platform is relatively suboptimal for like different organs in the mice. So I start a year long of the optimization of the EasySky from the very upstream of the mouse tissue dissection to the very end stream of this process to amplify the library. And after years of the efforts, we really developed this one optimized protocol for recovering the transcriptome from the all mammalian tissues, which not is only inherent from the previous EasySky that have a low cost, high throughput. But this protocol is really labor efficient that enable like a single person to finish most of the experiments. And I'm gonna demonstrate the other advantages in my following talks. And once we develop this platform, we are trying to apply this platform to study the organismal aging. And we learn from the previous studies that we really require a sex balanced cohort and need a multiple replicate. So we get the mouse samples from like 5 different life stages, and each stage will have like a 2 to 4 replicate for both sex. And we're also including 2 immunodeficiency gene mutant that really help us to dissect the cellular regulatory network in the aging process. And after several months of the experiment, we really established this PanSci atlas that in one study, we studied 13 different major mammalian organs spanning 93 different individuals. We profiled over like 600 samples. And in this one data set, we've been recovering the cell over like 20 million cells. And here I'm showing you the cell number for each individual organ and our initial clustering result of all the wild type cells. And you can see clearly that the cell from the different lineage normally cluster together. More than this data set really, for the first time, we can annotate the mean cell type, not at the full dataset level, but at each individual organ level. And after like a manual annotation and cross-validation of our annotation, we be annotate over 200 mean cell type across different mean organs. And there's two unique advantage of the PanSci dataset is that we really have a minimal batch effect, which is essential for us to identify the aging-associated cell types, because we are not just trying to build a single cell atlas, but we want to really identify the difference in the of the cell proportion between different age, and we don't want this difference to contribute by the batch effect. And I'm showing you here is like the batch effect between different experiments. We spec in the same kidney samples in the 15 different independent experiments. And I subject it to the dimension reduction and UMAP dimension reduction. And you can see that between different experiments, there is no clear batch effect. And on the left or right side, I'm showing you like different individual replicates in the same experiment. And we still do not see a clear batch effect in this individual replicate. Another advantage of the PanSci dataset is that we're leveraging, as I demonstrated before, that EasySky gave us a full gene body coverage that provide us both gene count information and exon count matrix information. And I'm trying to combine these two different layers of the information for the subclustering analysis. And here I'm showing you that for each organ, we annotate the mean cell type, and for each mean cell type, we zoom in and we use both gene and exon information to identify the subpopulations and maybe that may be interested for the aging discovery. And to summarize the first part, that we generate this PanSci dataset that define the 7 different we annotate the PanSci dataset at the full dataset level to annotate the 7 different lineage, and zooming into different organs, we annotate different mean cell type, and zooming into different mean cell type, we further identify the subpopulations. And now we are trying to ask what's the effect of the aging on those heterogeneous cell populations. And here I'm showing you one example in the muscle. We are trying to identify the cell population changes in the young and age group, and trying to find if there a significance, if there a cell type that underwent a significant depletion or enrichment in the aging. And let's just focus on the triangle here, that each triangle represents a mean cell type and its fold change. And we can see at the mean cell type level, most of the cells do not change a lot during the aging. But once we zoom into the subcluster level, which denoted by this like a circle, and we can see at the subtype level, there's a lot of cells population that underwent a significant change in the aging. And here I'm showing you one example about this Merck positive cells, which is very rare in the muscle and already identifies population changes at the mean cell type level. And previously people have already identified this like myonuclei subtypes that in the muscle, but people really do not go further to check its function or how aging affects these populations. And given our data set have a sex balance cohort, we can really check the cell proportion changes over the age in different sex. And surprisingly, we find these cell types underwent dramatic depletion in the aging, but this dynamic seems only happen in the female. In the male, we do not see a detectable or significant level of the decrease. And this partially can be explained by the marker gene of this cell type that specifically enrich in this locus on the maternal allele, which express a cluster of the microRNA that control the mitochondrial biogenesis and reaction to the reactive oxygen species. And this potentially implies some sex difference for this specific myonuclei to respond to the aging stress. And then we apply the similar analysis, not only in muscle, but to all the main cell type we identify in the PanSci dataset, and out of 239 main cell type, we can identify about 3,900 subpopulations. And by comparing the different time point, we pinpoint like about 280 aging-associated subpopulations. And in my following slide, I'm gonna focus on those aging-associated subpopulations. And we ask how aging affect those cells. And we really, the aging theory have been already established, can be since like 19th century. The very famous theory of the aging is this wear and tear theory, that body cells gradually deteriorate over time due to the repeated use and increased stress. And we ask, like, does this accumulation of the stress and damage over age affect different cell types in a similar way? To answer this question, we really take these 280 aging-associated subpopulations at different age. And here, in this heat map, each column represents each aging-associated subpopulations, and each row represents different age. And we subject this matrix to a hierarchical clustering analysis, and at the same time, we annotate each subpopulation with their lineage. And we can see that there is a clear cluster that clear cell population dynamics pattern over the age. For example, I'm showing you here at the early age, those cells accumulate in the young animal and they deplete in the aged animal. And we check their proportion of the cell lineage. We find that they are dominant by the adipose lineage and epithelial cells. And on the other hand, when we move to the late stage of the life, we see a dramatic expansion of the cells, and this expansion of the cells are dominant by the immune lineage. And this is very interesting, and we really want to zoom in to see what's happening for the immune cells. And leveraging the power of the PanSci, we have a very big data set with like minimal batch effect. So we can really extract all the immune cell from different organ and annotate the different subtypes of the immune cells. And here I'm showing you T cells that we extract from different organs, and we identify over about 18 different subtypes of the T cells. And once we do the differential abundance test, and we identify this CD4 positive naive T cell that is specifically depleted in the aging. And we also identify like 6 different expansion T cell subtypes in the aging. And I'm showing you one example here, this CD8 positive cytotoxic T cell, which have a marker of the GZMK. It's already been identified before as a conserved inflammaging markers. At the same time, we also extract the B cell and plasma cell from different organs. We've been successfully identifying 9 different subtypes of the B cells, and we also identify like 5 different subtypes of B cell that underwent dramatic expansion in the aging. And one example here I'm showing you is this, like we find most of the B cell aging-associated B cells or plasma cells express this IgM. And we know that IgM is a first response antibody to the infections that mediates like the universal and less specific immune response, which also like corresponds to the low-grade inflammation that happen in the aging organism. And shifting from this topic, once we identify those aging-associated cell types, and we find like the lymphocytes is really predominant in those aging-associated cell types, we ask like can we really identify dissect the role of the lymphocyte and what's the regulatory network of the lymphocyte and other cell populations, and how this regulation really shapes the aging hallmarks in the aging organism. Then we use this cell knockdown strategy, uh, uh, which is I'm gonna give you an analogy. Like when we in the normal genomic study, if we want to study a gene A's function, we normally knock down the expression of the gene A. If we find a gene B that shows a increased expression, we may infer the potentially regulatory relationship between this gene A and gene B. And same here, in these immunodeficiency mice, we specifically knock down all the lymphocyte, and we are trying to ask if there are other cell type that responds to this like knocking down of the lymphocyte cell population. And here is a validation of our immunodeficiency mice that we can really see a dramatic decrease of all the lymphocytes in the immunodeficiency mice. And we really try to ask with this knocking down strategy, can we infer the cell regulatory networks? And again, we take all these aging associated subpopulations from both wild-type animal, but also like immunodeficiency animal. And we still subject this matrix to the hierarchical clustering, and we identify like 4 different patterns. And as you can see in the first two group, they show those cell population changes do not show any difference in between the wild-type animal and immunodeficiency animals. We define those cells as a lymphocyte-independent cell populations. And more interestingly, in the last two cluster, we see there is a difference of the cell population dynamics between the wild-type animal and the immunodeficiency mice. And here I'm giving you 2 examples. One is about the aging-associated urothelial cell in the kidney, and another is aging-associated interstitial macrophage in the lung. And we can see that in the wild-type animal, it undergoes a dramatic expansion in the aging, but this expansion is completely shut down in the immunodeficiency mice. And this suggests a potential lymphocyte in epithelial and the lymphocyte myeloid interactions. To summarize, in this PanSci study, we develop an organism profiling platform, and we generate this over 20 million cells atlas, over 600 tissues, and we've been using this atlas to study the sex difference in the aging. We're trying to identify the aging cell population dynamics, and we also identify like the key immune effector of the inflammatory aging. And also we do a first exploration using this cell knockdown strategy for cellular regulatory network inference. And for the dataset availability, you can, if you are interested in our dataset, you can either get it from the NCBI GEO. Also, like you can access this dataset on the Azimuth application or UCSC cell browser. And we really thank the Rahul's team and the UCSC team for helping us to make this dataset more accessible to the broader scientific community. And in the end, I really want to spend one more slide to talk about the new challenges and the new opportunities that we are facing. That with more and more data we are gathering, and with more and more single cell technology have been already developed, we are entering this new era that we really need to see the biological question from different angles. In the conventional functional genomic study, people use all different high throughput sequencing platform to characterize aging expression. And here we really develop this platform to not only look at the gene, but look at the cell level And with this like high throughput platform, we can statistically significantly like perform multiple replicate for us to make like draw some conclusion. And we are also like trying to developing some like technology method to study the cellular regulatory network, and some recent some lab in the field also developed some like experimental validation for tracking the cell-cell interaction. And in the end, with all this like targeted cell perturbation method developed in the field, we really want to use this like functional cell omics tools to bring the cell type specificity to the conventional functional genomic study, the molecular hallmarks identified there, and to achieve a precise medicine for the healthy aging. And with that, I want to acknowledge my lab for their like tremendous support, without whom I cannot just finish this project on my own. And I also want to thank for the CICA lab and the UCSC cell browser to really help us to build the website and to make our data really accessible to more and more people. And with that, I'd like to take any questions. Thank you.
All right, that was great. Thank you so much. I really enjoyed the forward thinking bit of how the field is changing. So we'll move on to our Q&A now. If anyone would like to ask a question, you can raise your hand or type your question into the chat. And we'll start with a question. How did you unify the cell type annotation across different organs? Are you using the same marker genes for each major cell type in different organs?
Thanks for the question. I actually have a slide for this. So yes, we annotate each mean cell type on an organ by organ basis, and we first manually annotate each cell types with a published marker, and then we validate each mean cell type annotation with two approach. One is we train a support vector machine model to do the intro data validation, and we also cross-validate our data by mapping our aggregate transcriptome to the tabulary senescence data set, and we find like most of the cell like precisely mapped to the tabulary senescence.
Great, thank you. We have another question here. Akshay Varu, are you able to unmute?
Hi, yes, yeah. So I might have missed this because the journal is slightly late, but I see you're comparing the cell population dynamics across multiple time points. But have you looked at how do you exactly differentiate senescent cells versus aging cells? Is there specific transcriptomic or epigenetic markers that you use, or how do you define? Let's say if I have cells from 10 patients, everybody is the same age, but still you will have some kind of heterogeneity even after having being extracted from the same age. So how do you account for the heterogeneity in that?
Thank you for the question. If I understand correctly, that you're asking if some like replicate from, even they've come from the same age, they still show some like heterogeneity. And I agree that aging is a process that we do not know the end outcome of the aging. But here we are trying to identify the conserved aging cell population dynamics. That's to say that we only define a cell population that is aging associated by looking at all the replicate. If there is a statistically significance, between the replicate, then we say it's aging associated populations. But I agree that for the future study, we also really need to look at some like cells populations that only specifically enrich in like a specific individual, but not like all the individual from the same age. That could also be some like interesting way to explore the data set.
Thank you.
Great, thank you. Okay. Well, with that, thank you again for your talk. It's been very exciting. The seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. You can also follow us on Twitter at @CICE_ICA or check our website for the latest announcements regarding upcoming talks and events. And I'd like to thank everyone for joining, and we hope to see you in our next seminar. Have a wonderful day.
Thank you.